Hello everyone, I'm Hongda Wang from Professor Oscar's lab in the Electrical and Computer Engineering department. I'm greatly honored today to share with you our recent research progress, deep learning-enabled cross-modality super-resolution in fluorescence microscopy. To begin with, I'd like to first introduce some background knowledge. Here we show three major techniques of modern super-resolution fluorescence microscopy. These are stimulated emission depletion microscopes, structured illumination microscopes, and localization microscopes. So these are great tools that provide us with unprecedented access to the inner working of cells and also allow studies of various biological processes. However, they are not perfect. For example, they often rely on sophisticated optical setups and sometimes require extensive computational reconstruction. Furthermore, they introduce thousands to hundreds of thousands folds of more photon to the sample, which introduce an effect we call phototoxicity or photodamage. That is extremely bad for lab samples. So here we want to demonstrate a deep learning-based method that takes a single low-resolution image as input, enhance the resolution, to match the image quality that is captured from a more sophisticated setup. The first example is demonstrated with wide field imaging. So here, these images were captured with 10x objective length. Although the 10x objective has a larger field of view, the resolution is not ideal. A pre-trained neural network takes this image as input and super resolves it to reveal the fine structures, which is matched very well with the image captured with a 20x objective. We can also apply this method to confocal imaging. Here, this is a confocal image of keystone 3 size distribution using HeLa cell nuclei. Because this confocal system is a diffraction limited system, there's not much information can be revealed by these images. The network takes this single image as input, super resolves it, and matched well with the ground truth image, which was taken with a STAT microscope. The STAT microscope actually introduced thousands times more optical power for excitation and depletion. We can also draw the cross-section profiles to reveal this high degree of matching. Next, we applied our method to total internal reflection fluorescence microscopy, which is also called TIRF. Here, the object we studied is clathrin-coated pit. It is this kind of dome structure formed underneath the plasma membrane. When we are doing TIRF, we only illuminate the parts that are very close to the plasma membrane. So they should appear as donut shape. However, if you look at these TIRF images, because of diffraction limit, there's no inner structures within each donut. The network took these images as input, enhanced the resolution, and again matched with ground truth images, which was reconstructed from 9 TIRF images with structured illumination. Even in the smaller plaques or the small clathrin-coated pits that's spinning off a large plaque are very well resolved. In fact, this approach is so robust that we can apply it to a movie. Here we show the comparison movie of the network input, network enhanced image, and the ground truth image from 9 structured illumination movies. It stops on some frames that reveal significant biological processes. In addition to resolution improvement, there's an additional advantage of using a low NA lens. That's the extended depth of field. If you look at here, this is a blood vessel in the mouse brain. It's growing. It's a 3D structure that's growing out of the plane. So although the 20x objective gives us high-resolution image, the end of this blood vessel is already out of focus. However, that's not a problem for the 10x lens, although the resolution is not ideal. The network takes advantage of both worlds, gives us a high-resolution all-in-focus image from the 10x input image. To confirm the correctness of this network inference, we captured a Z-stack image of this blood vessel using the high magnification 20x lens and synthesized a extended depth of field image. The details are meshed very well, which again confirmed the network is revealing true structures. So at last, I would like to summarize our deep learning-based super-resolution framework. The deep neural network takes a single image as input and transforms it without iterative process. We demonstrated this method by low to high NA wide-field microscopic image transformation, confocal to STAD image transformation, as well as turf to turf seam microscopic image transformation. Our approach is purely data-driven, which means we do not need the knowledge of the image formation model, and in fact, such model is sometimes impossible to build. The network inferred image also has extended depth of field, as demonstrated with the blood vessel. We also observed that a pre-trained neural network model can generalize to new types of samples that the network has never seen before. At last, I would like to acknowledge all the funding agencies and collaborators for supporting our research, and thank you for your attention.